Hi, my name is Thompson. I'm a bachelor's student from the GRU Mobile Lab in Lawrenceville. I'm presenting this paper review with Angela Zaki, a master's student from the main GRU campus in Augusta. The paper we're presenting is molecular blood typing augments serologic testing. It allows for enhanced matching of red blood cells for transfusion in patients with sickle cell disease. Our objectives: by the end of this presentation, the student will be able to evaluate the importance of matching sickle cell disease patients with appropriate blood products. The student will be able to explain the methodology of molecular array genotyping and how it's used to predict human erythrocyte antigen phenotypes. And the student will be able to differentiate between serological and molecular phenotyping human erythrocyte antigens of sickle cell disease patients regarding cost, accuracy, and red blood cell component availability. The student will be able to distinguish between the different match categories presented and critique the advantages and disadvantages of high-level matching compared to mid-level and basic-level matching of red blood cell components and sickle cell disease patients. The student will be able to explain the inverse relationship that exists between the determined number of negative antigens and the number of available compatible blood components matched at the highest level. The student will be able to critique how evaluating a patient's GATA mutation status can impact the number of available red blood cell components matched for the patient at the highest level. And the student will be able to justify how molecular genotyping SCD patients using molecular array can lead to highly antigen-matched blood products. Alrighty, so first off, I'd like to give you all a brief overview of the sickle cell disease process with some statistics from the CDC. Sickle cell disease affects approximately 90,000 to 100,000 Americans. Sickle cell disease occurs among about 1 out of every 500 Black or African American births. Sickle cell disease occurs among about 1 out of every 36,000 Hispanic American births. Sickle cell trait occurs among about 1 in 12 Blacks or African Americans, and sickle cell disease is a major public health concern. From 1989 through 1993, an average of 75,000 hospitalizations were caused by sickle cell disease in the United States, and the ultimate cost was $475 million. So, RBCs are initially normal when a patient has hemoglobin S. They start out normal with a normal biconcave shape, and they're capable of deforming and changing shape and basically doing all the things that a normal red blood cell does and fitting through tight vasculature. However, repeated exposure to deoxygenated conditions will promote the characteristic sickle cell shape due to hemoglobin S forming precipitates and reducing a red blood cell's ability to return to its biconcave form, and instead forms the characteristic sickled form, which becomes irreversible. This form does not allow the red blood cell to pass through organs and tight vasculature and can lead to the systemic microvascular occlusions and many other health issues associated with the disease. The only real way to treat the disease is to supplement with transfusion therapy to ultimately reduce the ratio of hemoglobin S and restore oxygen carrying capacity. Now, the gene defect is a known mutation of a single nucleotide and is a single nucleotide polymorphism, or SNP, of an adenine to a thymine of the beta-globin chain, resulting in a polar amino acid, in this case glutamic acid, being substituted by a nonpolar amino acid, valine, at position 6. This is a benign mutation when oxygen concentrations are normal. However, when oxygen concentrations decrease, the deoxygenated form of hemoglobin S exposes a hydrophobic patch on the protein due to the valine, and ultimately the hemoglobin S molecule aggregates and forms fibrous precipitates that will sickle in deoxygenated environments. Repeated sickling of the hemoglobin reduces red blood cell membrane elasticity, and these rigid blood cells do not deform like normal red blood cells when passing through narrow capillaries, leading to vessel occlusions and ischemia. The bone marrow is not able to compensate by creating new red blood cells at a fast enough rate to match the destruction of the red blood cells through intravascular hemolysis. Sickle cells will only survive about 10 to 20 days, opposed to the typical 90 to 120 days a healthy red blood cell lives. The treatment for this condition is to reduce the hemoglobin S ratio in the body by transfusing non-hemoglobin S blood and restoring oxygen carrying capacity to the body. Some more background information. Antigenic variation exists between sickle cell disease patient phenotypes and primary blood donor population phenotypes, which is typically Caucasian. Sickle cell disease patients are more likely to develop alloantibodies due to available blood component products being comprised largely of dissimilar phenotypic cells, as well as an increased need for transfusions. The study also included a survey of several North American hospital transfusion service laboratories and found that approximately 2/3 of the hospitals surveyed do not determine red blood cell antigen phenotype for sickle cell disease patients with a negative antibody screen beyond ABO and D. They also found that approximately 1/3 of the hospitals surveyed will use additional testing extended to CE and CAL. The reasoning for this is that there currently is not a standard for hospital transfusion service laboratories as of 2011 at the time of this article being published to preemptively screen and match. The study was performed at the Puget Sound Institute of Pathology in the Puget Sound Blood Center in Seattle, Washington. 70 subjects were selected according to the following inclusion criteria. These were sickle cell disease patients who had been genotyped using the microarray human erythrocyte antigen bead chip platform and had the diagnosis of sickle cell disease and had undergone automated red blood cell exchange between 1999 and 2009. The study's hypothesis was such that additional information gained from molecular typing of sickle cell disease patients would improve the ability to find highly antigen-matched red blood cell components for transfusion support. The materials and methods used by the study basically included the antigens typed for being extensive and including clinically significant antigens such as big C, big E, little e, CAL, Kid A, Kid B, Duffy B, big S, and little s. Molecular added a benefit over serological testing of also detecting Duffy silencing mutations and Duffy null phenotype mutations, or GATA. So basically, the study utilized whole blood and DNA extraction kits from whole blood, such as PureGene DNA procedure. Cells were lysed using an anionic detergent along with the DNA stabilizer to reduce degradation of DNA in the presence of DNA present in the environment. Protein contaminants were removed using precipitation. And genomic DNA was recovered by adding alcohol and precipitation. PureGene procedures ultimately use a modified salting out precipitation method to purify DNA. The main system used by this study was the BioArray HEA bead chip, which is a high-throughput molecular assay capable of detecting 38 red blood cell antigens and several phenotypic variations. As you can see, it lists a couple of the extended antigens that this system can detect. So they use the DNA gained from the DNA extraction kits and combined it with the master mix for single tube multiplex polymerase chain reaction amplification. So DNA isolation is performed using whole blood and a blood DNA extraction kit. During the amplification, specific DNA segments are selected and amplified using multiplex PCR or PCR with multiple primer sets, which produces several different amplified regions of DNA of various sizes so that several sections can be detected using one procedure. The PCR products are processed to remove any residual primers, and then single-stranded DNA is generated. The amplified DNA will bind or anneal with a specific oligonucleotide probe on its target bead. When a perfect match is formed, the probes will elongate. Once elongation has occurred, the target bead will fluoresce, and a specific bead fluorescence profile is created and analyzed depending on which beads have elongated. The bead fluorescence profile is translated using BASIS, or Bioarray Solutions Information System, a software available from the company to laboratories, which will interpret and translate the genotype and phenotype interpretations based on the output of the assay. Basically, this study utilized a liquid red blood cell inventory query for each sickle cell disease patient's predicted red blood cell antigen phenotype. The sickle cell disease patients were matched based on their ABO compatibility and negative status for minor blood group antigens that the patient lacked, depending on the match level. The study created 3 different match level categories. The basic level included ABO, D, big E, big C, little c, and Kel matching. The mid-level included ABO, D, big E, big C, little c, K, Duffy A, and Duffy B matching, and high-level antigen matching included ABO, D, E, big E, big C, little c, Kel, Duffy A, Duffy B, Kid A, Kid B, Big S, and little s. And they also included sublevels for whether or not a patient was positive for the GATA mutation or not. Mid and high-level match categories included the subcategory for patients homozygous and heterozygous for the GATA mutation. Patients who were found positive for the GATA mutation had their blood component search expanded to include Duffy B positive and negative blood, since these patients should rarely make anti-Duffy B. And now I'd like to transition the presentation over to Angela, who will talk to you all more about the specifics of the GATA mutation, results of the study, and discuss the practical implications.